Student, good morning. To continue the lecture, linkage. Morgan's view on linkage. Morgan stated that the pairs of gene of homozygous parent tend to enter in same gametes and to remain together, whereas same genes from heterozygous parents tend to enter in different gametes and remain apart from each other. He further stated that the tendency of linked genes remaining together in original combinations in due to their location in same chromosome, the degree of strength of linkage depends upon the distance between the linked gene in the chromosome. Morgan's concept about the linkage developed the theory of linear arrangement of genes in the chromosomes, which helped the cytogeneticist in the construction of genetics or linkage maps on chromosomes. Chromosome theory of linkage. The chromosome theory of linkage of Morgan and Castell states that the genes which show linkage are situated in the same pair of chromosomes. The linked genes remain arranged in a linear fashion on the chromosome. Each linked gene has a definite and constant order in the arrangement. The distance between the linked genes determines the degree of strength of linkage. The closely located genes show strong linkage. Then the widely located genes which show weak linkage. The linked genes remain in their original combinations during the course of inheritance. Arrangement of linked genes. When two pairs of genes are linked, the linkage may be of either of two types in an individual heterozygous for both pairs dihybrid is to dihybrid. The number one, the two dominant genes, example capital R and R0 of sweet pea, may be located on one members of the chromosome pair with two recessive genes R and R0. On the other, or the dominant alleles of one pair and the recessive of the other R and R0 may be located on one chromosome of the pair with the recessive of the first gene pair and the dominant of the second gene R and R0 on the other chromosomes. The first arrangement with two dominant on same chromosomes is called as cis arrangement. Cis arrangement. This type of interrelationship of dominant and recessive linked genes, example RR0 oblique RR0 is called as coupling phase. The second arrangement, one dominant and one recessive gene on the same chromosomes is called as trans arrangement. Such type of interrelationship of dominant and recessive genes. Example R, capital R, small r, O, oblique R, small r, capital R, O is called repulsion phase. Cis and trans arrangement of two pairs of linkage genes of Lathyrus odoratus after the Braun in 1969. Kinds of linkages. The phenomenon of linkage is following two kinds: complete linkage and incomplete linkage. Incomplete linkage. When the linked genes are so closely located in chromosomes that they inherit in same linkage groups for two or more generations in a continuous and regular fashion, then they are called completely linked genes, and the phenomenon of inheritance of completely linked genes is called complete linkage. Example: according to Bridge, all the genes of male Drosophila remain completely linked. Further, in a mutant state of Drosophila, the genes for bent wings B plus and 7 Bristol SVN of the fourth chromosome exhibit complete linkage. Second is incomplete linkage. The linked genes do not always stay together because homologous non-sister chromatids may exchange segments of varying length which bearing many linked genes with one another during meiotic prophase. By the process of crossing over, the linked genes which are widely located in the chromosomes and have chances of separation by crossing over are called incompletely linked genes, and the phenomenon of their inheritance is called as incomplete linkage. Incomplete linkage has been observed in PG maize, tomato, female Drosophila, mice, poultry and man. Here the example of linkage have been considered only for Drosophila and G maize means maize, maca. Incomplete linkage in Drosophila. Wild type Drosophila has grey body. Wild type Drosophila has grey body and long wing B plus and V plus, oblique B plus V plus, where alleles for grey B plus and long V plus dominate over the mutant alleles for black B and vestigial V. When a grey long fly B plus V plus oblique B plus V plus is crossed with a black bodies and vestigial wings fly BV oblique BV. The F1 heterozygote is found with grey long phenotype and B plus V plus oblique BV genotype. The F1 heterozygote B plus V plus oblique BV when test cross with double recessive parent BV oblique BV, instead of occurring of two class of phenotypes in the ratio of 1:1, occurs four classes of phenotypes as shown in The test cross results are clearly showing that parental combinations gray long and black vestigial are those expected from complete linkage and appears in 83% cases. The other two gray vestigial and black long are new combinations and appears in 17% cases, thus in 17 cases crossing over the occurs. Second is incomplete linkage in maize. In C maize, in case of incomplete linkage between the alleles for color and shape of seed has been observed by Hutchison when a maize plant with seeds having color and full endosperm CS oblique CS capital C capital S oblique capital C capital S is crossed with another plants having recessive alleles or colorless shrunken seeds small c small s oblique small c small s. The F1 heterozygote are found with the phenotype of colored full and genotype of capital C capital S oblique small c small s. When F1 hybrid is test cross with double recessive parent small cs small cs. Diagram showing the cross involving linkage and crossing over the genes for vestigial versus normal long and black versus gray body in Drosophila are linked. They are located in the same chromosomes. Classes of descendants are obtained instead of two as shown in the figure. Colorful cross with colorless shrunken capital C capital S oblique capital C capital S small s cs oblique small cs. Colorful capital C capital S oblique small c small s. F1 colorful cross with colorless shrunken capital C capital S oblique small c small s small c small s oblique small c small s. Test cross results colorless colorful colored shrunken colorless full colorless shrunken. 48 capital C capital S oblique capital C capital S 48%. Capital C small s oblique small c small s 2%. Small c capital S small c small s 2%. Small c small s oblique small c small s 48%. The test cross results are the clearly shown the parent combinations of the alleles. This is the kinds of linkage, complete linkage and incomplete linkage. Okay, thank you.